Hello, everyone. My name is Leah, and I'm Bay Nature's event coordinator. I'm really grateful to you all for joining us this at noon today, and I'm really excited to welcome you to today's Bay Nature talk on seals of the Farallon Islands. Before we begin, I just want to encourage everyone to turn their cameras on if you're comfortable so that we can all be together for this hour. But please keep your mic muted unless you're asking a question. Before we begin, I'd like to introduce Bay Nature magazine really quickly. Bay Nature is a quarterly print magazine that's based in Berkeley, and it focuses on nature and conservation topics that are local to the 9 counties of the Bay Area. In addition to our print magazine, we also publish weekly digital-only content and host a number of events like these talks, as well as in-person hikes and other events. So if you're interested in receiving free access to these hikes and talks, I encourage you all to become Bay Nature members, and I'll circulate a link for that later. But without further ado, I'm really excited to introduce our three presenters today, starting with Meredith Elliott. And Meredith is a principal scientist at Point Blue Conservation Science and has worked on a variety of seabird monitoring and diet projects. She supervises the Point Blue Marine Laboratory, where data on marine food webs are collected, and is a program leader with the Access Program, an at-sea monitoring program founded by Point Blue, Greater Farallons National Marine Sanctuary, and the Cordell Bank National Marine Sanctuary. Thank you, Meredith, for joining us. And then I'd also like to introduce Amanda Spears, who is a Farallon program biologist with the Point Blue Conservation Science Group, whose work involves supervising long-term seabird research on the Southeast Farallon Island during the spring and summer. And this monitoring informs management of local marine ecosystems in the face of climate change. Amanda joined Point Blue in 2018, and through this experience, discovered her love for seabirds and the California current ecosystem. And then thank you so much, Amanda, for joining us. Jeannie is our third speaker today. I'd like to invite Jeannie on screen, and Jeannie Johnson is Point Blue's Marine Lab assistant who processes samples from the Farallon Islands and access cruises. Jeannie graduated from UC Santa Cruz with a bachelor's degree in marine biology and has studied manta and devil ray bycatch mitigation for her senior thesis. Jeannie loves getting people excited about science and enjoys learning about and contributing to conservation efforts to save threatened species and restore ecosystems. Thank you all three for joining us today, and I'm going to turn it over to Amanda first to present. So thank you all.
Hi, thank you, Leah. Yeah, it's great to be on today, coming from you live from the outdoor classroom of the Farallon Islands, including virtually. Yeah, it's wonderful to talk to you about our pinniped species today. Those are seals and sea lions. And so I just want to share my screen here. And bring up some fun photos and learn about some seals and sea lions today. All right. So this is our Let's Talk Seals program. And so first I want to talk about what are seals and sea lions, right? Maybe you've seen them yourself before. And in the Bay Area, they are sea mammals alongside with cetaceans, or you know them as whales and dolphins, also manatees and dugongs, and also sea otters. And these animals evolved from weasel or bear-like animals that lived on land, but then entered the sea approximately 33 million years ago. And evolved and diversified through that time to give us these wonderful carnivores that live a kind of dual existence, and that's the seals and sea lions. And I may be referring to them in this talk quite frequently with the word pinniped. And we use that word a lot in research with these animals. And pinniped has a cool Latin meaning, pinna meaning winged or feathered, and then ped being foot. So these are the flipper-footed or wing-footed animals. And yeah, what is the difference between our seals and our sea lions? So you've got two groups, essentially. You've got your earless seals, and those are the true seals, or the phocids. And out here on the Farallon Islands, We have two species of phocids. We have the northern elephant seal and the harbor seal. And the difference between them and our sea lions is that they don't have any external ear flaps. They do have ears and they can hear, but you can't see the external parts of the ear on the outside of the head. And then they also have these small front flippers, which are really good for maneuvering in the water and then also for crawling up onto land. So they're also known as the crawling seals. But primarily they use their hind flippers for swimming. So when they're in the water and they're swimming, they use their flippers almost like a fish's fins. So they sway them side to side and, really efficient swimmers in that way. But they can't really walk on their hind flippers. They can't turn them forward and stand. They have to move kind of like a caterpillar on land. And I guess the technical term for this is galumping. And maybe you've seen this in harbor seals videos or in person where they're just kind of inching their way forward across the land. So it can make them quite slow on land, but they are incredibly fast in the water. These animals, these true seals, tend to be more quiet and less social, both on land and at sea. Now comparing that to our eared seals, which are our fur seals and our sea lions, these are grouped into the otariids, and they are recognizable by their external ear flaps. So you can see their little ears. I don't know if you can see the little external ear flap there on the side of this California sea lion. They tend to have large, strong front flippers, and these act almost as oars in the water and produces most of the propulsion, forward momentum in the water. So super strong. And they also can rotate their hind flippers forward out of the water to make them agile and quick on land. So these are they're the California sea lions. You can see them kind of marching along on the land. It makes them really good climbers as well. And these guys, the otariids, they tend to be pretty loud and definitely more gregarious and social animals. And so, in relevance to the pinniped species that we study out here that are present in the California current ecosystem, those 5 species are the northern elephant seal, the Pacific harbor seal, the northern fur seal, the stellar sea lion, and the California sea lion. And maybe you've already been able to identify which ones are the true seals, our earless seals, and which ones are our eared seals or our seals and sea lions. So the phocids, the earless seals, are the northern elephant seal and the Pacific harbor seal. And then our eared seals, our sea lions, includes northern fur seal, stellar sea lion, and California sea lion. I wanted to show you some videos of these animals on land. We have the privilege of researching these animals on a daily basis, monitoring their populations, monitoring their productivity through their breeding season, and being able to watch them in their in their home, in their haul out areas, in their rookeries, their breeding areas. So this is an example of a California sea lion haul out. And you can hear that there's lots of barking. This is very important places for them to rest during the day, after their foraging bouts at sea, as well as for avoiding predators that lurk in the waters, being it killer whales or great white sharks. So these animals are relaxing on the beach in this big old cuddle puddle. You could see some of the, immature animals kind of wrestling in the foreground. These are immature males. We call this thumb wrestling. But there's also some adult females in here, some adult males, some pups. And so they really love this beach area. This is North Landing, but they're incredible climbers as well, climbing in these rocky slopes and doing, just super well out here on the Farallons. Definitely our most numerous pinniped species that we have out here. And then moving to the next one. Ah, forward, forward. There we go. I am primarily out here during the spring and summer months, which is the breeding season for California sea lions. And really, the Farallons didn't become an important breeding ground for this species until about 2 decades ago. Very few pups were born on the islands back in the early 2000s. And, now at this point, we have, over 12 to 18,000 animals that we count on the island. Over 1,000 pups are born here. And this is kind of an example of how cute they are. And this is taken from a blind. So the animals cannot see us, but they are very curious. I also wanted to introduce you to the Steller sea lion. They haul out and pup on the Farallons in much lower numbers. I mentioned the 12 to 18,000 number for California sea lion. Steller sea lion, approximately 100 to 200 animals during the peak season, which is also during the summer. This is a video of a female, an adult female, calling to her pup of the year. And so they do this a lot too, a very busy area, very large island. Mom goes out to forage during the day. She comes back to rest and needs to find her pup. So they often communicate with one another to find each other. And she's letting this pup know that it's time to nurse. Really cute example of that. These animals, the Steller sea lion, a little bit difficult to identify, distinguish from California sea lion, but they are much larger. They're more bareheaded, very square head, very much lower voice than, what you heard in the previous video, the barking, dog-like barking of the California sea lion. This one is very low and more of a roar. You can kind of understand why these animals are called sea lions. Yeah. And then this is a bull, Steller sea lion, just kind of relaxing in the intertidal. Very large animals. And they can weigh up to, a ton, just over a ton, these big bull males. Resting. And these animals, a lot of these pinniped species, they exhibit polygamy. So they, will have a harem of females that a single male protects. And that's really, due to, this being a very concentrated place for these animals to breed. And so you see this in the northern fur seals, you see this in the stellar sea lion, the northern elephant seals, and definitely in the California sea lions as well, but a little less so. I've heard them be referenced as kind of more free love type, but yeah, that can lead to this amazing size difference between the males and females. Here's your northern elephant seal bull males. Really cool video of them kind of squaring off, showing off their big noses, their size. These adult bull elephant seals often have significant scarring on their neck areas from battles. Pretty intense. And then here are some immature animals. And they sound really funny. So I wanted to include that. So this is from activities, working in the elephant seal breeding colony. These animals are less wary of the researchers here. And so, we're able to get a little closer, look for tagged animals, be able to read those tags, and monitor their survival. So I wanted to kind of wrap that up and say, what are we doing on Cephi to research pinnipeds and conserve our pinnipeds? So a lot of that is monitoring the population productivity of all of these seal and sea lion species. And that's through weekly counts that we do, as well as, pup counts, how many pups are these animals producing in a season, and really diving deep into survival and individual known age productivity in our northern elephant seal colony. We're also, there's animals that are tagged or branded, either here or at other locations. San Miguel, the Privlofts are some examples where we can recite individuals to get a better understanding of where they're breeding, where they're hauling out, where they're moving in the system as a whole. We're also documenting entanglements, and I'll be talking about that a little later. But, these animals living out in the open ocean, going for fish, they are at risk of being entangled in fishing gear and marine debris. And then we are sometimes getting to rescue baby seals. This is a photo of some researchers during a gnarly winter storm where a elephant seal pup was washing away. And so they were bringing it kind of out of the swell zone and back to safety into the colony. And then most importantly, potentially, is our collection of scat samples. So these animals, lots of animals hauling out in these rocky areas, and they're eating and they're pooping. So that's a way for us to understand, potentially what they're eating. And so, in the field, we go out and we collect that scat, and then we send it off back to the mainland, where our team at the Marine Lab goes into a little bit more of the analysis and processing of those samples. And so that's kind of where I want to end it on the field end for the pinnipeds on Cephi. And you can take it away, Jeannie, with the lab portion.
Alrighty. Thank you so much, Amanda, for that explanation. So yes, so continuing on to my what I do here in the lab is yes, so we get our samples from the Farallon Islands and then we wash them through a process with a washing machine and a portable washing machine, and we put them in a mesh bag. And so that way all the actual poop gets washed away, and what we are left with is a bag full of bones. And we go through that bag of bones and we look for otoliths, which are fish ear bones. And so now I will turn on my microscope camera. And so this is a real life California sea lion scat sample. And so this is what we look at. It's always a toss up about what we will find in here, so it's a little mystery. And this one was very rocky. So sometimes the sea lions just eat a lot of sand because their faces are very close to the ground. And so this guy actually has some parasites. So these are lungworms. These are just parasitic worms that we find in our sea lion samples, and we always make note of it. And that was about all the things that I could find. But yeah, so we just sort through this, and then what we are interested in, sometimes we find fish vertebrae or other like spiny fish bones that are like part of their ribs or something like that, or their skull. But what we are interested in is their inner ear bones. And so their inner ear bones are called otoliths, and we care about those because I like to call them a microchip of information. And so they can be used for lots of different things in science. What we use them for is they are different shapes for different based on different species. And so what we do is we look at the shape and we are able to identify what species of fish our predators are eating. And so that tells us, one, how well our predators are eating, like our sea lions. And then it also tells us what kinds of fish and the population sizes of the fish that they are eating. So it kind of gives us like a whole ecosystem thing. Lots of information about the ecosystem and the food web. And so what we do is, I will show you now. So in this sample, we actually found, so these bigger otoliths up here are actually from the fish called Pacific hake. And so they're much bigger and wider. And then down here, these little guys with the pointy tips, those are northern anchovy. And so northern anchovy are one of our most common fish that we find. They are very high in protein and have fatty oils that are very nutritious for our predators. And so what we do is, so just like how we each have a left and right ear, so do fish. And so we side them left and right. And so we put them on our sulcus side. So our sulcus is our, sorry, I'm just trying to fix this. Okay, so our sulcus is this line that's like right in the middle. So if you see this little groove in there, that sort of tells us that we are on the right side, and that helps us orientate it. And then we have these pointy bits. And so this pointy side that's sticking out is called the rostrum. And so that helps us side our otoliths and tells us that there's a left side and a right side. And then that tells us that because there's a left and right side, that means that our sea lion ate one fish, and then we're able to count the number of fish that they ate. And so then after we side and ID them, then we put them in our database, and then we see what kind of fish they're all eating. And then another way to see what kind of fish they eat is by looking at their scales. So with scales, we can't tell how many prey they're eating because fish have like numerous scales. It would be like counting the skin cells. So what we do instead is we just look for different kinds of scales. So bony fish mainly have two types of scales. They will either have cycloid scales, cycloid scales, which are what northern anchovy have. And that is what is pictured here. Let me just zoom in on a scale. And so these cycloid scales are They have annuli and a focus, and they're not very radial compared to ctenoid scales. So ctenoid scales have a thing called a radii, which kind of looks like a scallop shell. It has these little wavy grooves in it, along with a focus and a circular annuli. And so those look more like fingerprints, where these anchovy scales are more, they kind of look like monstera leaves, if you can find one. And so we use that to just sort of see what prey are present. So if we find a Pacific hake scale, which would be a ctenoid scale, then we know that it at least ate a Pacific hake, because sometimes we can't always find the otoliths. So it just sort of backs up and gives us a broader idea of what kind of prey they're eating. And sea lions are very opportunistic, so they don't only eat fish, they also eat squids and octopus and whatever else they might find in the ocean. And so right here, what we found in this sample is actually this is a market squid beak, and this is their top beak. So each squid, just like how we have a top jaw and a bottom jaw, squids have a top beak and a bottom beak. So this is a market squid beak, and we can tell it's a top one because their top beaks look more like a raptor's, like a bird beak, and so they're very sharp and pointy at the tip. And market squid beaks are usually clear on the inside. And normally, if you were to look at a squid, you would only be able to see that little black tip. And this clear part underneath is what's attached to the inside of the squid. And so this is their, like, the center of their mouth, and this is how they eat their prey. And then what we also found in this sample is I believe this is a bobtail squid. And this is also a top beak. And so this is just another type of squid that we find in our samples. And I think that is just about it. And so now I will pass it off to Meredith, who will tell us what we do with all this data and all this prey information that we find. And yeah.
Great. Thank you, Jeannie. Yeah, thanks for showing everybody what we do in the lab, all that painstaking work to identify all the little things that the California sea lions have been eating and poop out. Yeah, I'll just start by saying this is a great way, a great non-invasive way to look at what sea lions eat. We can get a good idea of the species composition and numbers of different prey that the sea lions are eating. And over time, that can give us a really nice picture of how these different prey populations might be changing over time. So yeah, so maybe I will start by just sharing my screen, which so I promise I'll only show one graph. It's kind of nice to see the videos and the microscope work. So I'm only going to show this one figure. But this is showing basically our diet results since we started this work back in 2014. So as Amanda mentioned, these scat samples are collected quarterly, so they're kind of give an idea of what the sea lions are eating in different seasons in the year. And so the x-axis is showing the year and the season. And then the y-axis is showing the percent by number. So what that means is out of all the prey things that Jeannie and others in the lab have been identifying, you know, what percent of those prey we have identified are these different groups of prey. So I've done my best to simplify it. We have 6 different groups of prey shown here, which make up 80% or more of the diet in any given collection. So those include the cephalopod, the squid and octopus. Jeannie showed us those beaks. So those are shown in the gray bars on these graphs. The Pacific jack mackerel is shown in yellow. Rockfish is shown in that orange color. Then we have northern anchovy shown in that dark blue. We have flatfish shown in the bright green teal color. And then a Pacific hake shown in the pink, which Jeannie showed some good examples of anchovy and hake otoliths. So you got a sense for what those look like. So, you know, I'll first start by saying that that all these groups are fish that humans fish for, either commercially or recreationally. So, you know, when we think about the broader context of our diet work, of all the monitoring that happens out of the Farallons, putting this all together, we can kind of get a sense for what the current primary threats are to seals and sea lions, as well as kind of thinking ahead to what some of the impending secondary threats to these animals might be. So, you know, knowing that all of these prey are also fished by humans, there's going to be some interactions with fisheries. And Amanda touched on this by showing the picture of the sea lion in the entangled in fishing gear. Seals and sea lions are also perceived as being in competition for these prey with fishers. So yeah, these interactions with the fisheries are of concern. Also, climate change is a big concern for these animals, as it is and is going to bring changes to the prey that they rely on. So, as Amanda talked earlier about, you know, the pinnipeds here are really adapted for cold water conditions. With the Farallons, we're right in this great upwelling center where we get these cold, nutrient-rich waters. But with climate change, that's expected to change. We might see more, you know, those warm water conditions, that we might see more frequent and more intense El Nino or El Nino-like conditions, where we get these warm tropical waters from the more southern latitudes. Also, in 2014 to 2016, maybe some of you have heard of the North Pacific marine heat wave that we had off our coast. We had very warm waters during that period. And as you can see in this figure, during those years, which are kind of on the bars more on the left side of the graph, the diet was pretty diversified. Like they ate a lot of the different groups of prey that are shown here. As we transitioned out of that marine heat wave, that's where we started to see maybe bigger numbers of the cephalopods, greater numbers of the northern anchovy that really returned to their diet after those warm water conditions kind of faded. So yeah, those warm waters change the distribution of many of the prey that they rely on. So northern anchovy was known to expanded their distribution northward during the heat wave. Market squid, one of the cephalopods they eat a lot of, also did that. So yeah, our diet work is kind of helping to see these changes in the prey. You know, marine predators are great indicators of what's happening in the environment. So, so maybe I'll stop sharing my screen now. And yeah, someone actually talked about domoic acid. Yeah, and I think Amanda may, I'll turn it over to you in a second, Amanda, but yeah, so yeah, currently there is a domoic acid event happening along the Central Coast, down by San Luis Obispo area. One of our colleagues, Julie Hauer, is very involved in observing that. And yeah, so as those of you may or may not know, domoic acid toxicosis is a big problem for marine wildlife. This domoic acid happens when we have these harmful algal blooms. There's a particular phytoplankton species called Pseudonychia that produces a neurotoxin that can then accumulate in the fish that pinnipeds and other marine wildlife depend on. So that's of a big concern. We typically see those events in warm water periods, especially in the fall. So yeah, there is one along the Central Coast. There may be indications that it's starting to travel up north, but I'll pass it off to Amanda now, and maybe she can touch on that.
Sure. So, domoic acid is something that we see occasionally out here on the Farallons. Now, really to be able to identify it, I think with any certainty, excuse the Western gulls, they're quite rude. But with any certainty, you need to do necropsies on animals. And so, anecdotally, with the event happening in areas, south, we have had a few animals in the last 2 weeks, I would say, maybe 10 or so that we've seen die on the island. I haven't seen any animals that are alive showing active symptoms of domoic acid, but we have had a few mysterious deaths in the last 2 weeks. So we're continually monitoring that situation. And if we do see, significant increases in mortality of particularly California sea lion, then that's something that we would get our partners with NOAA and potentially the Marine Mammal Center involved with to be able to identify and say, yes, this is domoic acid. In regards to some of the other secondary, risks and threats that these pinnipeds face, that we, is very relevant to our work on the Farallons. Again, I showed that photo of a California sea lion with an entanglement. And, primarily these entanglements are of monofilament fishing net. And, it's unclear whether or not that's from, an active gill netting operation or it's, these quote unquote ghost nets that are basically abandoned or, lost fishing gear. But regardless, they get entangled with this gear, and then it is typically around their necks or around their bodies. A lot of the ones with the neck entanglements can live and continue kind of their daily activities, but it is something that as it's exposed to more sunlight, it tends to tighten up, it cuts into the skin, and can lead to really nasty infections. And I've seen lots of sea lions be able to survive with incredible wounds, like, missed shark predation attacks, but, something like an entanglement where something's kind of slowly, deepening and embedding in the neck. I have seen animals die from entanglement injuries. It's also really difficult to disentangle a sea lion. We are not qualified out here currently to be able to sedate large animals and disentangle them. But there are people in the area that do, and the Marine Mammal Center is one of them that deals with entangled animals, which is great. And then often they release those animals, sometimes near the Farallons, and they have a little tag on them, and we can see that they are an animal that's doing well and thriving on the Farallons post an entanglement. So other things are island habitat disturbance. I mean, just disturbance at all in their, places that are really important to these animals on their, whelping ground or their pupping grounds, their resting areas, their breeding grounds, their rookeries, any sort of disturbance. These animals, do not like people generally being, close by. So, every time a pinniped enters the water, it's at risk of predation and just, you're expending more energy, right? Kind of going back and forth. So, if you remember, how you feel after a long day swimming, you can probably imagine how these animals feel when they're resting on land. Another thing, is low genetic diversity. I mean, that's something that we don't really, dive into out here specifically with our monitoring, though it is an issue that impacts, has impacted northern elephant seals, because of, and potentially northern fur seals because of their near extinction and complete extirpation from these islands during the fur trade, fur trade time period during the 1800s. And then pollution. So, there are pollutants that enter our waterways, PCBs and DDT, that, I was reading actually with the Marine Mammal Center, they've, potentially identified as carcinogens or cofactors with the herpes virus that is causing increased instances of cancer in California sea lions, upwards of 20% plus of their stranded adult animals that they see in their rehab facilities. And then also microplastics, and that's kind of a realm that we've been diving into and hope to continue with in the future through our scat collection. We're able to, hopefully process those scat and look at potential microplastic exposure to these animals. It's a growing concern. It has been found in the bodies of marine mammals, and it has been shown to impact growth rates and fertility in small fish and zooplankton. So that's kind of on the cusp. Yeah, those are some of our things on our radar when we're talking about management and conservation for these animals. And then Meredith, if you want to talk about, the smart management that comes out of the work that we do out here, that would be fantastic.
Yeah, I'll just keep it brief because I know we're getting a little short on time. But yeah, ultimately our diet data can help us understand not only what the pinnipeds are eating, but also our hope is that our data goes into contributing to what's called ecosystem-based fisheries management. And this is just a way of thinking about fisheries and recognizing the ecosystem and species interactions. So by combining the diet data with the population data from the Farallones and other rookeries along the California coast, we can make better estimates on basically how many metric tons California sea lions are consuming of these different prey, and try to provide those estimates to fisheries managers like the Pacific Fisheries Management Council, and can help them make more informed decisions about how much we should take while leaving enough for the marine wildlife. So yeah, I guess I'll end it there. I don't know if Leah, you want to take it over from here, or
Oh yeah,
yeah.
we have. Thank you all three. Before I just want to thank you all for sharing, and it's such a cool glimpse to see, in the field and then in the lab and then kind of broader impacts. That's such a neat view, and I appreciate you all sharing with us. And we do have a lot of questions, so let's see how many we can get through. First, I'd like to try ask Mary McAllister. You had one question. Would you mind unmuting and asking your question? And I can also, if you prefer, ask your question out loud.
Can you hear me?
Yes, I can hear you.
I asked about whether or not the seals were present on land on the island just during breeding season or year round. And now that you've shown graphs subsequently, I'm guessing the answer is that they haul out onto the islands throughout the year. Is that correct?
Hi, yes, that is correct. For the northern elephant seals, their breeding season is from December through February. which is kind of a flip from our
Yeah.
odorides, our sea lions. But they are, each species of the 5 species are hauling out and present on the island year round. It's just that they're, at any given time, the number changes, and it tends to be seasonal in relation to their pupping and their molting.
Thank you.
Cool. Our next question is from Elisa. Elisa, would you mind unmuting and asking your question? I can also ask. I think Elisa's question was, how many elephant seals are on the Farallons?
Yeah, so currently, right now it's the summer season, and this tends to be the lowest amount of elephant seals that we see. I think I counted 5 in Garbage Gulch just the other day. So it's like, below 20 this time of year, but increases steadily through the fall season as, a lot of the immatures come back to molt. And we've seen a few of the bulls come back to molt as well. And then, cows really start showing up later, like, November time. And, our breeding population is just kind of, a couple hundred individuals, not even 100 cows at this point. We have seen declines in our elephant seal colony. And we think due to increased intensity of winter storms, that is just kind of degrading the optimal beach habitat where they like to pop. But they are increasing, in colonies on the mainland, which is good to hear.
Yeah, no, that's awesome. Thank you for that. And let's see, I think another question from Bridget. Bridget, would you like to unmute and ask your question?
Hi, yes. I was just wondering if there is a way that you guys estimate the age of reproductive adult seals on the Farallons and how like the tagging program plays into the age estimation.
Yeah, so we aren't aging them specifically to, year, unless it's an individual that we know and tagged as a pup, in its first year. So we have a solid understanding of some of the known age individuals for the elephant seal colony, but we haven't and currently do not tag puffs of California sea lions or stellar sea lions. But we do tagging of our northern fur seal pups in the fall. So I'm less familiar, honestly, with our understanding of returns of like fur seals to the colonies and at what ages. But I know when we're, doing our population monitoring, these animals reach, sexual maturity, for females and males, it differs, but they tend to show, morphological or physical differences that you can identify, a male versus a female, and what might be, an adult female versus an immature animal. And that kind of gets into a little bit more of the finer details of identification of animals. Yeah.
Thank you, though. Thank you for sharing that. And it looks like we have a lot of questions from Yvonne. And Yvonne, it sounds like it's best if I ask, so I'll go ahead and read your questions out of the chat. So, well, first question is, are there modifications to the cervical and thoracic spine vertebrae that strengthen and help protect the male northern elephant seals during their seasonal battles with each other?
That is a great question. I don't know. Honestly, that's something that I'm going to have to look up. But certainly I can figure out who can answer that question, and we can, have a back and forth via email. Meredith or Jeannie, do you know anything about the skeletal system of northern elephant seals?
I don't, but it's got to be pretty solid to take the impact, huh? That's a great
Yeah.
question. I don't know.
Yeah, I mean, what the only thing I can speak to of that is, the adult male elephant seals can reach upwards of, over 2 tons. So there must be something structurally sound about them.
Yeah, awesome. And then another question from Yvonne was, in Yvonne's experience, fur seal rookeries smell so different from sea lion rookeries. And is this due to differences in diet or from differences in their skin and fur oils? And can you, like, just for me, I was wondering, could you describe what it smells like to be there? And like, yeah, what is it like to be there?
Yeah, personally, I'm less familiar with the elephant seal rookery smell. I think that's the specialty of our winter biologist, James Yost, but I know that their scat is quite different. Their scat of the elephant seals tends to be pretty like viscous and orange. And certainly they are foraging at different depths than some of our California sea lions, stellar sea lions, and on different prey species. So I think that's a great guess to us, kind of make about it potentially being related to diet. But the California sea lions, yeah, it kind of reminds me of like a mix of like cow manure and dog scat. It's quite stinky. It's very stinky. It's very inspiring to watch them and how free they are in relieving their bowels kind of wherever and whenever. So yeah, it is definitely an intense smell that luckily we get nose blind too, unless it's a real fresh one.
Awesome. Thank you for sharing that. And Yvonne had another question about, in cases where there was a death due to domoic acid, what would the necropsy show? Like, what are things that could indicate that that was the cause of death?
Well, I know domoic acid leads to like heart failure and seizures in the brain. I haven't personally done a necropsy. I've kind of seen one done on an animal before, but basically they're taking certain organs like and probably brain tissue and looking at kind of signals of that neurotoxin being inside their body.
Gotcha. Thank you for that. And let's see, there was one question for you, Jeannie, actually, about your educational background. And yeah, would you mind sharing what type of educational background prepared you for your role in the lab?
Yes, yeah. Yes, I think I replied to Claire, but yeah, I went to undergrad at UC Santa Cruz, like you said, and I did my undergraduate on manta rays and double rays in the tuna fishing industries that are caught as bycatch. And along with that, I took a comparative vertebrate anatomy class, as well as ichthyology class and an invertebrate zoology class. And so that'll help me just sort of identify different species. I actually got to extract otoliths from a fish, and that was very great. And along with that, yeah, I just really liked fish, and my passion is actually sharks. So I've done a lot of my own research as well, and I've also just had Meredith and my old coworker Natalia, who are great at teaching me and doing training me on the job to understand how to go through all these samples. And so quite honestly, I don't think I could have done it without them. And Meredith is an excellent teacher and is very patient. And so I was able to understand everything because of her and all of her knowledge. So yeah.
So cool. And I think one, yeah, I think one really great last question actually is from Lishka. Lishka, would you like to unmute and ask your question about Farallon conditions that began to attract the predators to these islands?
Oh, sure. Can you hear me?
Yes,
Okay.
can you? Well,
Great. Yeah, Meredith, I know the story about, or maybe I forget who brought this up. Maybe it was you, Amanda, but maybe you can both answer. But I'm familiar with the fur seal story and how hunting sort of decimated that population. And when the island became a refuge at some point in time, the fur seals were able to come back and their numbers are rising. But I wasn't as familiar with the California sea lions, and that was a new thing for me that they had arrived at the island about only 20 years ago. So I was just wondering what were the conditions that changed that attracted the California sea lions to the Farallons.
Yeah, I don't know what attracted them 20 years ago. I do know that in 2014, when we started our diet study, there seemed to be an influx of California sea lions then, and that was due to the marine heat wave. I don't know the conditions 10 years before that. I guess it was another that we were starting to get into an El Nino-like situation, but 2004 was a pretty, I guess, normal year, if I recall. So yeah, I don't know, Amanda, if you have any insights on what happened then.
Yeah, so I do want to clarify that California sea lions have always had a presence out here on the Farallons. But they really didn't start pupping like in earnest until like in the last like 15, 20 years. So there were a few pups that, would be born, less than 50. And then, now like this year I counted over 1,200 pups. And that's been just kind of this exponential increase in the number of pups we've had born on Seppi. And, if I had to guess, I would say that it's probably, they seem to be doing really well in responding to the amount of northern anchovy in the system. That seems to be something that caught my eye. Just looking at the diet data and our increase in numbers of sea lions pupping, as well as just immature animals falling out. So, distance to food, abundance of food, certain particular foods like northern anchovy most likely play a big part in that increase.
Awesome, terrific. I think we will wrap it up there. We're at the 1 hour or 1 p.m. mark. And I just want to thank you all again so much for being here, for showing us the 3 really different but so integrated perspectives of the work that you do. And yeah, it was just really exciting to get to hear from you all and see your guys' presentation. So thank you so much. And thank you all for joining today and listening. If you're interested in, again, free access to these talks, please consider being a Bay Nature member. I'm going to send a link out to everybody with the recording from today's talk, as well as some links from our hosts. And just want to thank you all again for joining and hope you have a great rest of your day. Thank you all for joining.
Thanks, Leah.
Thanks, everybody.
Thank you.
Yeah, Leah.